Thank you. Welcome to AP Biology. I am going to try to give you all the fundamentals of chapter 23. This is going to be a long video / lecture, so please remain focused. Remain focused. This chapter is about how evolution occurs on a smaller scale. And we're going to look at a term in a second called microevolution. So we're looking at all the mechanisms that exist, or the fundamental mechanisms that exist, that help explain what would change a population even from one generation to just the next generation. And your book starts out in the chapter talking about an island in the Galapagos Islands, or in the Galapagos Island chain, called Daphne Major. And there have been scientists there studying the bird populations and taking a lot of population data from the birds that live on this island for decades now. So we have a lot of good data about every year about what the bird populations look like, primarily the finches, which is a type of bird. And what they discovered back in the 70s was that during a severe period of drought, the beaks of these populations began to show small changes. And they were able to point out how that was related to environmental changes. So here's what they found data-wise, or a little snippet of their data. What they found was during this severe drought, the drought-tolerant plants had larger seeds, and that these birds' food source was predominantly from these large seeds. And the birds with the bigger beaks had beaks that were capable of actually eating those larger seeds, which made up most of the food source during this very difficult environmental period. And so before the drought and then after the drought, comparing the data, they found a small increase in beak size. Now, we have many points of data like this in populations all over the world where we see an environmental pressure lead to a change in a population over just a few generations, a fairly short period of time. But for the purposes not just of talking to you about evolution, I do want to take a second and talk to you about data representation. Because this is important not just in science, but in politics and everyday life. I want you to start getting a little bit more comfortable with some of these real-world application of statistics. Those two bars are really different, aren't they? Contrasting colors. One's like almost twice as tall as the other one. Look at that huge difference. What is the difference in a unit of length between these two averages? Like half a millimeter. This is a good example of how you take, I can't even do this and show you half a millimeter. It would be like, that's half a millimeter. Except my fingers are not steady enough to not occasionally touch each other. It's hard to hold your fingers even half a millimeter apart. But we have instruments that can measure fine lengths, and the data did show, I mean, that's a fairly substantial increase. We're talking about, a 5% increase in beak length. That's not nothing, and they have enough data to make it a significant difference. But this is just in general a good example of how you can take some really small difference and make it look like a huge difference. But getting back to the point, we can track small changes in populations from one generation to the next. And these small changes are what we refer to as microevolution. In the 1970s, on this island, we didn't see birds undergo some kind of unbelievable change where they hardly looked like they looked earlier in the decade. No, it was a small, roughly half-millimeter shift in average beak length of the birds on this island. That's a tiny change, but you would expect it to be tiny because it was over 3 or 4 or 5 generations. That's not many generations. So that's what we refer to as microevolution. Now there's a term here that I have rarely used because we just haven't gotten to the topic yet, and that is this term right here, allele frequency. That's what we've really learned to look for when we look for evolution in populations, changes in populations over time. Allele frequency. You could also, instead of using the word allele, use the word gene. A gene and an allele, they're very similar terms. Gene's more general, allele is like the flavor of the gene, whether it's the dominant one or the recessive one. But we look for how common a gene or genes are in a population over the course of time. If those numbers are changing, then the population is changing, because a population and its traits are going to be linked to their genes. So, we're going to focus a lot in this chapter and today on gene frequency or allele frequency, and we're going to look at eventually how natural selection and then these other two terms, genetic drift and gene flow, affect it. So we're going to look more at all of the big points here. We're going to start by talking about allele frequencies here in a second, and then we'll hit on these other points as well. So it's all about how common genes are. Okay? So that leads us to this concept of genetic variation. How much variety exists? How many different alleles might you have in a population for a specific trait? That is what we call genetic variation. Now, there's a lot of ways you can define it, but in general, genetic variation is very good for a population. If you have a lot of variation, you want to think of that as being a good thing. A more popular phrase we would use today is like diversity, but genetic variation really hones in on the actual gene diversity, the genetic diversity. Now, there's really only one source for getting a brand new, never before seen allele. What is it? Mutation. You have to have a change in your DNA to get a new segment of DNA that can do something new. So mutation is very important when we talk about new traits arising and expanding upon genetic variation. But there are other ways to bring about variation in populations, and through sexual reproduction, you have one of those ways. So with sexual reproduction, 2 different individuals can shuffle the genes they have. Now, without mutation, these are genes you already see in the population, but by shuffling them, you get different gene combinations, which increases variation. And you typically get things like crossing over from meiosis, that can happen in sexually reproducing organisms. So genetic variation is important. You want to have as many sources of different alleles for a trait as you can have in your population. And why? What is good? Why is it better to have a population? Why is a population more extinction resistant if you have variation versus if every individual is a clone of every other individual?
Better at adapting.
You have a better chance of adapting to a changing environment. If we were all clones of one another and the environment changed in a way that could kill one of us, it might kill all of us. So variation at least allows some members of the population a chance of surviving, if those variations work out to be beneficial. All right. On the topic of reproduction, what we find a lot of times is how you reproduce can affect variation. We've talked a little bit about this. If you have a very rapid reproductive rate, meaning maybe every hour, every few hours, every day, you're creating a new or the next generation, you can really accumulate a lot of mutations and variation in that way. We see that happening with the COVID-19 virus, where this is such a fast reproducing, I don't want to say living thing because viruses don't technically fit into the definition of life, but it's such a quickly reproducing entity that mutations show up very frequently, and you start getting that new strain. That's all part of its kind of genetic variation. We reproduce very slowly compared to most living things. Takes us 15, 20, 40 years to produce that next generation. That's a lot different than every hour. But the advantage of sexual reproduction is that you have things like crossing over, mate selection, you randomly pass on one chromosome or the other. There's a lot of ways to get variation, even if you don't reproduce fast, but usually it requires the benefits of sexual reproduction. Kind of two different strategies. I think y'all are going to be impressed with how much I got to fit on this slide. Look at that. No margins. So, gene pools. Again, on the topic of genetics, and there's a lot that we'll pull from Unit 5 when we talked about genetics a few months ago. Gene pools, when you hear that term gene pool, that refers to all the genes, all the alleles for a trait that you can find in an entire population. And I have a little definition for population up there, but I think you have a fairly basic understanding of what a population is. So you take a group of organisms that are interacting and interbreeding with one another, and you look at what the genotype is for every individual, and then you just add up or account for all of the genes of all of those individuals for a trait. So if it was a population of pea plants, you could see how many big T's and how many little t's you had in totality. So we have to look at the whole gene pool to get an idea of what the gene frequencies are, which gives us an idea as to what direction evolution is pushing us in, or pushing that population in, and how quickly. Okay? So as much information is written up there, that's really all I have to say about it, but I would recommend you read over some of this later. So to give you an idea of the gene pool and how we calculate gene frequencies, we're going to go back to this example of red and white flowers that blend through incomplete dominance, blend to make a pink heterozygote. So here we see some numbers. We got a population of 500. They have the number of red, pink, and white flowers. And you're either red, pink, or white because you have some combination of two genes. So two red alleles, and it's a red flower. Two white alleles is a white flower, and one of each, the heterozygote, would be the pink flowers. Now, if you have a population of these flowers, then you can simply just count how many of each phenotype you see. And in this case, every unique genotype has a unique phenotype. So every possible combination, you can see red, this combo is pink, this genotype, this combination is white. So out of 500, we can figure out percentages or frequencies of certain phenotypes. That's actually called a phenotypic frequency. You did some of this with Punnett squares when you wrote out the Punnett square and you guessed that, there might be a 3-to-1 ratio or a 75-to-25 breakdown of certain offspring. Well, we could just look at a population and take these frequencies. Now, in order to figure out information about specific, not individuals, but the genes those people are carrying, you have to keep in mind that how many genes is a plant, in this case, going to have for this trait? How many alleles would you have per individual for this trait? 2! Yes, 2. So this population of 500 would have a gene pool of 1,000 genes, 2 for each individual. Okay? So if you count up the numbers or proportion of just the red allele or just the white allele, you can go through and use this information to figure out what we would call an allele frequency. So if we have 320 individuals that are red, that's 320 individuals that have 2 reds. So 2 times 320, plus we have 160 heterozygotes. One of their alleles is red. So 160 of them, they have 1 red each. You had 160. That's 800 out of 1,000 alleles that are the red allele, which is what percentage? 80%. Or if we say it as a frequency, you drop the 100%, and it's just 0.8. Now, what do your percentages have to equal up to? 100%. So obviously if 800 out of 1,000 are red, or 80%, the rest of them are the other allele. So those are the kinds of numbers we look at when we track populations to see how they are evolving and why they're evolving. So on this slide, they do some of the math for us. So here we have 800 out of the 1,000 possible genes is a 0.8 frequency, or 80%. And then you have the rest being 0.2. Now, the variables P and Q are variables we're going to use in actual math problems. So make a note of these. P and Q are the variables for the dominant and the recessive alleles. P is for the dominant allele, Q is for the recessive allele. and they have to equal 100%, or when we deal with frequency as opposed to percent, 1. So 0.8 and 0.2, or 80% and 20% is the one whole pool that we're dealing with. So it's not that hard in, simpler examples with simpler populations that work on simpler genetic principles. It's not too hard in the fundamental examples to find actual numbers that you can track. So this population has 80% red genes in the gene pool, 20% white genes in the gene pool. If you come back 50 years from now and look at that population again, if those numbers have changed, that's change over time, which is evolution. So that's all you have to look at is the allele frequency to determine whether a population has evolved. Now, again, evolution doesn't mean that these flowers are going to evolve into crazy new life forms. It's microevolution. We can track these small changes over small periods of time. And we do that with an equation called the Hardy-Weinberg equation. This is where the math for this unit will be. So I'm going to show you the equation in a slide or two. And you will have to use it to solve for certain variables. Thought somebody was getting tired. I don't want that. So this Hardy-Weinberg equation is based on a model that we can use to track evolution in a population, even if it's just small changes in the population over shorter periods of time. And there's a concept in this model called the Hardy-Weinberg equilibrium, which we will talk about in just a second. Now, one thing I want to mention is that I thought I just saw the word locus. You see this word locus a lot when you talk about Hardy-Weinberg mathematics. Locus, basically, it would mean location or spot on a chromosome. And it's kind of a fancy way of just referring to a gene, in a sense. Every gene is found at a certain spot on a certain chromosome. So if you see that, you just want to think about, like they use this phrasing, alleles for a given locus. That just means the possible different genes you could have at that same spot on the chromosome. That's all that means. So this Hardy-Weinberg equation uses P and Q for the dominant, and I guess in this case they're not dominant recessive, but for one gene versus the other, typically dominant or recessive. This is incomplete dominance, so you don't really have that. And it uses this idea of a gene pool, okay? So in our flower, red, pink, and white flower example, we calculated a frequency of 0.8 for the red genes and 0.2 for the white genes. And you can imagine your gene pool as like this pool right here with genes in it. And when you make a new individual or you create the next generation, you just reach in and grab 2 genes and you have your new diploid organism. All right? So it's based on that kind of thinking. That right now, the human population is made up of people with genes. All those genes are our gene pool. And the next population will basically come from genes in this generation being pulled down or whatever, carried on to the next generation. Now, if you take it in a very kind of literal, I'm pulling genes out of a hat way, the mathematics works just like any other statistics or coin tosses. So if you have 80% of the genes being for the red flower, 20% of the genes in the population for a white flower, then if you randomly draw from a gene pool that's 80% red, 20% white, you can calculate what you would expect to see. Expected data. So if you have an 80% chance of drawing red, then in homozygous red individual, the odds of getting that next generation are just the odds of pulling a red gene times the odds of pulling a red gene, which comes out to p squared. All right? You could do this with coin tosses, except it would be 0.5 and 0.5 for heads and tails. Same math, except we're using 0.8 and 0.2 because our example had 80% and 20% for the breakdown of the gene pool. Well, 8 times 8 is 64, or 0.64 of a frequency. You would expect this population, if nothing was affecting it, to produce 64% red individuals. Now let's look at the white flowers as opposed to the red. The white genes are 20% of the gene pool, or 0.2. If we square that 0.2, we get 0.04. 4% of this population, next generation, should be white flowers. It's a little bit weird for the heterozygotes because you can get a red and a white, or a white and then a red. So the value is calculated using 2pq, which in this case is 32%. So this is what we would expect to see next generation if the gene pool is 80-20 red genes to white genes. But do populations reproduce and survive in a random, just pulling it out of a hat way? Do they?
No.
No, they do not. They do not. So your book talks about how you can apply this to like a Punnett square format, but we're not going to worry about this. I don't want to spend too much time on this topic. What I really want you to focus on is this equation. P squared plus 2pq plus q squared is 1. This is the Hardy-Weinberg equation. There's a great chance there will be a question about it on the AP exam. And you will have questions about it on the test. These variables actually mean something. This isn't like a math class where it's just letters and numbers and you don't understand what it means. That was a joke. These letters mean something. P squared in this equation should represent the homozygous dominant. Q squared should represent the homozygous recessive. And the 2pq part should represent heterozygotes, one of each gene. Well, unless you have some complex genetics going on, which we're not dealing with, we're keeping it like kind of more simple Mendelian examples. Your homozygous dominant, homozygous recessive, and heterozygous individuals should be all of them. So they should add up to 1. So that's kind of where you get this equation and what they mean. But if you take nothing else away from what I've said to this point, P squared is the homozygous dominant, Q squared is the homozygous recessive, and the 2pq represents your heterozygous individuals and how common they are. Now, like I said, using this equation, using these variables and the gene frequencies that we've talked about in the gene pool, you can get expected frequencies. These aren't numbers you're really going to see when you go out into the woods and start counting squirrels and looking at their traits. These are numbers you would expect to see. And based on what assumptions are we using? Well, that's where we have what we call Hardy-Weinberg equilibrium. That equation tells us what the number should be if the population is not evolving. How is a population not going to change from one generation to the next? These 5 conditions must be true. You will maintain a static, stable gene frequency from one generation to the next, meaning no evolution is occurring, if there's no mutations, if mating is completely a random affair, if the environment is not selecting for or against certain traits, if the population is big enough, I don't know what the critical value would be, and if there's no genes entering or leaving through migration, which we'll talk about gene flow in a little bit. By the way, do we know of any populations that all of this is true for? Just say yes or no. 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 No, we don't. All right? So this is a kind of hypothetical model, or a theoretical model. Now, what this means, you might think, well, if there's no population that ever fits these conditions and the equation's based on these conditions, then why use it if it never exists? We can still get an expected number of what percentage or frequency of each individual you should see in a population. And then when you don't see that in the real world population, you can start to see why. So just to go back real quick, we have counted this population. We know that point A to point 2 are the frequencies of those 2 genes. So we would assume next generation we would see this. Well, what if instead of 4% of the population next generation having white flowers, it was like 30%? We could learn something from that. We could say, there's only supposed to be 4% white flowers, but there's 30%. Somewhere, one of these conditions is not holding true. Where is it? Is it that it's an advantage to have a white flower? Is this a tiny population where these numbers are just wildly fluctuating, which is a problem in small populations? Are all the red flowers leaving for greener pastures? We can start to identify what's the cause of the change. Why are we not seeing what we expect? And that leads us to understanding why the population may be changing and what evolution is up to right now in that population. So that's how we apply it. We basically use it to see whether or not evolution is having some influence on a population and how and why it is. Okay, y'all been very patient so far, and we're making good progress. I'm going to throw some more vocab at you, just so you've heard it before and, I can give you a brief understanding of it. So I mentioned earlier, natural selection, genetic drift, and gene flow are 3 big factors that can affect how common certain genes are. which means they affect evolution. They affect how populations change. I want to look at these three. So we're going to look, I've already talked about it a little bit, but I'm going to look real quick at natural selection and then drift and flow. And this next slide, just list them again. That was a little bit more repetitive than I thought it would be. But natural selection first. So basically, the concept of natural selection is just that the environment the population is in is going to favor or not favor certain traits. Okay? That's all natural selection is saying. You put a population into an environment. Is that environment extraordinarily hot and dry? Well, there are certain traits that are good for surviving there. Is the environment cold and snowy? Well, there are certain traits that are going to be better there. And that's natural selection. The idea that nature, the environment the population is in, the environment itself can increase or decrease the genes you see from one generation to the next. Just takes a different set of skills to survive in the desert than it does in the rainforest or the grasslands. And that leads to what we call adaptive evolution. Essentially, the individuals that are best adapted to the environment are going to change future populations by surviving and having the children. Then you have some things that Darwin didn't really think about and that are more of a modern mechanisms that we've discovered. There is this concept of genetic drift, which is kind of a fun one. Basically, random things can just throw a wrench into the whole process. It kind of doesn't matter sometimes how well adapted you are to the environment. You could be the smartest, strongest, fastest, most handsome, best-looking individual in the whole population. And you could get struck by lightning before you have any kids and pass any of those genes on, and that's it. So sometimes it doesn't matter that you were well adapted. You still don't make it. Sometimes you're not well adapted for the environment at all, and through a series of very fortunate events or luck, you end up having lots of kids, and they're not very well adapted either. Hey, it happens. That's genetic drift, when you hear that term. It's a fancy sounding term for something that's basically just like, luck plays a role. Random things happen. That's genetic drift. Kind of within that idea, you have what's called the founder effect. The founder effect. And we see this. We've actually seen, since the publication of the theory of evolution, we have found like volcanic islands break the surface, life colonize them. This is something we've had enough time to observe to some extent. But you can have small segments of a population break off and go somewhere else to a new environment, like an island. And then, over many generations, that new place, that island that was founded by this small group, it starts to look different than the original population that those founders came from. So, the original individuals that show up in an area, they are the foundation for future populations. You can imagine, like, kind of a popular topic these days is the colonization of Mars. Let's say we were ready to go. And they took, people from this class and sent them to Mars. And that's who started the Martian population. And then let's say civilization collapsed on Earth, and 10,000 years later, we built ourselves back up, and we went to Mars, and we found these people, and we were like, whoa, there are people here. Who we sent to Mars originally would have a huge effect, genetically speaking, on what that Martian population would look like when we went back. I mean, if we sent a bunch of tall people, I could see it being a taller population of individuals than the Earth had, unless natural selection affected it in some other way. So it matters who kind of starts the new population, and some of that can be random. And in their example here, they literally talk about like indiscriminately blown to a new island. That's kind of what happened with the Galapagos. The Galapagos Islands are very weird, and we think a lot of the creatures and plants that got there just kind of like got blown out to these islands in a hurricane, or flew off course on some migration pattern and ended up at these islands and just started the populations there. That's the founder effect. That can have an effect on what the population looks like that really might not have much to do with natural selection. A similar idea is the bottleneck effect. This is also part of kind of genetic drift. It's kind of like the founder effect, except nobody goes anywhere. The founders in this case are just people but the individuals that survive a catastrophe. Humans seem to have gone through a pretty substantial bottleneck. And the idea of a bottleneck is you have a population, and something causes that population to be dramatically reduced in size. So if you track the population, it's kind of like a wide, large population. A catastrophe occurs, and then it narrows, almost like the neck of a bottle, is what it would look like if you graphed it out as a population chart. Now, after this great catastrophe, future populations are going to resemble the survivors. And we see this in the fossil record, we see this in genetic studies. Humans are surprisingly genetically similar all over the world. And what it points to is a time, we have some evidence for this, it looks like about 80 to 100,000 years ago, the region in Africa where humans were basically all concentrated back then underwent some severe periods of drought. So severe we can see evidence in the fossil record. And based on how genetically similar we all are, and the fact that that happened about 80 to 100,000 years ago, there are estimates that the world population of humans at that time dwindled down to maybe only 1,000 people. We may have just been 1,000 people on this planet, and we are all their descendants. That's a small group, an enormous bottleneck to go through.
Are there any Wills in here? People named Will?
No one named Will.
All right. Thank you.
Good luck with your search. Okay, then there's what we call gene flow. Gene flow is just essentially migration. This isn't you're blown to an island and start all over again. This isn't a giant catastrophe came in and wiped the population down to just a few survivors. Individuals just kind of leave and enter populations. and this is kind of where I want you to kind of think in terms of gene frequency. When a person leaves a population, they take their genes with them. And when a person enters a population, they bring their genes with them to the gene pool or out of the gene pool. So there is the chance, if enough individuals leave or enter a gene pool, that gene frequencies can change. All of these things affect gene frequency. Okay. The last few slides. You know what? This is the last section. Let's stop there. We can finish the